JJ and I are here again. We are going to do 3 kitchen labs today. The first one is in your Moodle is called, by the way, these are in week 2. The first one is called Membranes Alive. I have always, since I've taught biology I've taught for many years, I've always called it the egg cell lab. But so I might say that, sometimes, but it's called Membranes Alive. And you're actually studying the characteristics of the cell membrane, the plasma membrane. But today is day 1, and day 1 is super easy. All you're going to do is put your egg in a container and number that container 1, 2, 3, and 4. You're going to have 4 eggs. And all you need to do today is, if you have a kitchen scale, you do need to weigh each egg and write down the weight. I have a kitchen scale, so I'm going to do that. I'm going to show you how to do it. But if you don't have a kitchen scale, listen to me carefully. The purpose of this lab is not to find out the change in the weight. It's just to find out what happened to the egg after it sat in certain solutions. So you want to know if it got bigger, or if it got smaller, or if it stayed the same size. So really, you just have to have a way of measuring the size of the egg. So I'm going to show you, for those of you that do have a kitchen scale, what we're going to do is we're going to take the egg out, put the kitchen scale down, and sit it. Mine has just a button. It's already doing it. It says tare, so I just hit that button, and it's measuring the weight of my container as nothing. Yeah. Now I can put my egg in here, and it's going to tell me how many grams that my egg is. And it's 64 grams, so we do need a pen. That's something I got everything together, and I didn't get a pen. But the first one is 64 grams. And you should really redo this one, because this one is 3 grams lighter than the other container, even though they're the exact same kind. So you should re-zero it. Okay, that's 0 grams. Now I put the egg in, and it's 66. So that's why, if you're doing this precisely with a scale, you need to be precise. If you don't have a scale, what you can do is get you a ruler or a tape measure and do the best you can. Make sure you use the metric system and just measure the width of the egg. And your pictures can do a lot too. You can take pictures of the egg alongside of the ruler, so that you can see what it measures. And you can also take pictures of the egg, like up against some notebook paper, and that kind of gives you a visual of what it looks like. You just got to keep track of your pictures. You've got to know this is egg number 2. But this will give you a visual of about how big the egg was before we added vinegar. But the purpose of adding vinegar is simply, is that 66? Yeah. Is simply to, you can go ahead and do these and tell me the amounts. The purpose of adding the vinegar to the egg is simply to dissolve the shell. We want the shell dissolved so that the membrane underneath that eggshell can act as the plasma membrane, and we can see which types of substances move across the membrane, either into the cell or out of the cell. We're going to see a process called osmosis. So this is going to demonstrate that, but it's going to take a few days. What was this one?
Oh, you wrote it.
Let's try this one again. I believe you.
Yeah, I just put this one on here, but yeah.
Okay, I believe you. It's just that it's a big difference.
Well, I teared the thing. I wasn't teared with it. You know what I mean?
No, that's fine. It was correct.
Oh.
So now JJ is going to pour vinegar into these containers until the eggs are covered up with vinegar. And I'm going to start explaining to you the next lab setup that we're going to do, which is the photosynthesis lab. For the photosynthesis lab, it does call for you to have 4 syringes. So let me show you what I mean. I don't mean needle syringes.
Is this too much?
No, that's perfect.
Okay, make sure.
The amount of vinegar has to cover the egg. The egg must be submerged in the vinegar. It needs to get, because otherwise some of the shell will not come off. Okay, you see these syringes that little kids get, you know, whenever they have medicine, that kind of thing. We have a whole bunch at my house because we have 2 grandchildren, so it wasn't hard for me to get these, but you can get them at Walmart. But you do need a syringe like this, and your 4 syringes need to be the same size. You don't need to have one bigger than another one. They need to all be the same size. Mine is a 5 milliliter syringe, but I know they make 10 milliliter syringes and they make 2.5 milliliter syringes. So the first thing we're going to do is we're going to make our baking soda solution, and that we're going to make that by adding 1/4 of a cup, 1 1/4 of a cup of water to, I forgot, let me find it. It's the next page. I forgot how much baking soda, if it's a teaspoon or a tablespoon. It says 1/8 of a teaspoon, so we're just going to put a pinch, like we did last time, a little pinch of baking soda. Here's our glass, and I'm just going to go to the sink. I've got 1/4 of a cup. I'm going to put 5 of these in my glass. One, 2, 3, 4, 5, and then a pinch of baking soda. You can pour it out in your hand if you want to, but then just get a pinch, because this one's all hard. So I'm just going to get a pinch, what I can pinch in my fingers, and I don't know, just for good measure, I'm going to get 2 pinches. I can't really explain sometimes why I do things, but I've been doing this a long time. It just seemed like I needed 2 pinches. So, so anyway, then you just wipe your hands to get that baking soda off of there, and you can stir this if you want to, or swirl it. It's going to mix really fast. But what we're going to do is we are going to fill our syringes with that baking soda solution. 2 syringes will be just baking soda and little round discs that we cut out of spinach leaves. Because what we're going to do is measure the spinach leaves. We're going to use 10 spinach leaves in the syringe at the beginning of the lab, and we're going to measure for about 30 minutes, every 5 minutes or so, to see how many of those spinach discs rise up. And that's going to tell us something about photosynthesis. But right now I just want to show you how to do it, okay? Because there is a technique involved. Make sure you number these. I have for number 1, it's just going to be the sodium, the baking soda solution and spinach. And then number 2 is going to be same thing, baking soda solution and spinach, but I'm going to put it in a dark chamber. I'm going to keep one of them under, I've got a lizard in my bedroom that has heat lamp and also ultraviolet lamp. It is dark outside, so I'm going to do this in my lizard's cage, my bearded dragon's cage. I don't expect you guys to have the same stuff that I have, so just wait till it's light outside. You can just sit it outside. You can technically sit it in the light of your room, but ultraviolet light is best, so sunlight is best, okay? Then the other 2 are going to be 2 different colors of food coloring added to our baking soda solution, and we're going to put them in the light as well. So the one in the dark has no color in it, and then the ones in the light are going to be clear, and then 2 different colors of food coloring. It's Christmas, Christmas season. I know everybody doesn't celebrate it, but those that do, I got green and red food coloring, but it's not because of Christmas. Actually, it's because the green light and the green food coloring and the red food coloring will show us something about how, you know, which colors of light are best for plants. So we have now. I want JJ to move where you can see what she's doing.
Okay.
We don't have a hole punch. In the lab it says you have to have a hole punch. Let me show you what we did. We cut a big thick straw in half. This is a straw we used for last week's lab. And we're using the round end of it because it's hard. We're using that to cut little discs out of spinach leaves. Make sure that your spinach leaves are healthy and green. Lay them down. And you're going to need 40 discs. Now,
They look like this. Maybe you can
yes,
see.
JJ's going to hold it
Super
up.
tiny.
Yeah, so you can see it's about
Like
the
a
size.
little circle.
Yeah.
Tiny little circle.
What you want to make sure is that the discs are no bigger than the end of your plunger, because that's the diameter of what they're going to have to fit inside of. So you want them to be no bigger than the diameter of the bottom of your plunger. That's a good way to get the size. Because you can even cut these out with scissors. You can just take and cut little circles. They don't have to be perfect. But you are going to need 40. Do we have 40?
Yeah.
Okay, perfect. Okay, so what we're going to do is I'm just going to do this with number 1, and then I'm going to turn off the video because it's just the same thing over and over. But, okay, so with number 1, I'm going to put 10 spinach discs.
Should I go with this one?
I just want to make sure they can see. Can they see my hand?
Yeah, they can see.
That's 1, 2.
You need to bring the camera over here and hold it or anything?
No, just as long as you guys know I'm counting these discs. That was 3, 4, 5, because I'll bring it up there when I do the important part. 6, 7, 8, 9, and 10. Then we're going to put the plunger back in it. And we're going to draw up some sodium bicarbonate solution. I'll come up close to you. We're going to draw up sodium bicarbonate solution into this. So I have to push some air out for a minute. Okay, in order to draw up any solution. And I'm going to draw up as much as I can without pulling the plunger out. So here's what I've got. You see where the spinach leaves are? You see how they're floating on the top? That's bad. We don't want them floating on the top. So we're going to push the air up. We're going to push the air up. There's air above the spinach leaves. So, and some water too. But we're going to push the air out. And can you hold this for me,
yeah.
JJ? I forgot. Okay, and I'll just clean up my floor later.
Yeah.
But anyway, I want to push it until I see solution go all the way to the tip. Then I'm going to put my finger on it and pull on that plunger. You see what's happening? You guys maybe can see bubbles. I'm pulling the air out of those discs.
Wow.
Yeah. So now I want to push them back up because there's air. I want to get the air out of those discs.